Hi, welcome to this lesson where we're going to be reviewing the multiple choice questions published by the OCR exam board, which are on unit 2.4 on enzymes. Okay, so the first thing to do, if you haven't done these questions yet, is to follow the link in the description, do these questions, and then come back to this video when you're ready to mark them and see how you've done. Okay, pause the video now if you need to do that. All right, let's get going. So question one, which of the diagrams illustrates the way in which the rate of an enzyme-controlled reaction depends on substrate concentration? So that is diagram A. Okay, so initially it depends on the substrate concentration, but later on it doesn't, and something else like enzyme concentration might be limiting. Question two. When starch and amylase are mixed in the presence of iodine solution, so remember this goes blue-black, the blue-black colour rapidly disappears. This is due to the formation of which molecule? Okay, so we know here that it's digestion of the starch by the amylase, but what is actually the end result? I bet you might have gone wrong on this one because it's actually this. It's actually maltose. So what happens if I was to draw sort of starch as a long chain, whoops, it's supposed to be the same size, a long chain of little molecules joined together by glycosidic bonds like this. These are supposed to all be glucoses joined together as starch. The product is actually this. It's actually maltose. So maltose is a disaccharide like that. Okay, the product is maltose, and it is then further digested later on. Okay, so it's maltose. So amylase, salivary amylase in your saliva glands, digest starch into maltose, and then there's another enzyme later on in the digestive system in the small intestine called maltase that breaks down maltose into two molecules of alpha glucose. Okay, so that's something just to remember. It's a bit of a tricky one. Okay, moving on. Question 3. Which of the following enzyme groups can catalyze oxidation reactions? Okay, well, there's nothing here that's obvious, like an oxidase. So we have to think, what is equivalent to oxidation? So oxidation is loss of electrons. It's also loss of hydrogen. Okay, so which one is it? Loss of hydrogen is D. So dehydrogenase is the same thing as oxidation. Moving on. Question 4. So here we have concentration of substrate going along the bottom and rate of reaction here. So the enzyme concentration is kept constant throughout. That's important. The enzyme concentration is kept constant. At concentrations greater than X, which of the following statements is true? The rate of reaction is limited by enzyme concentration after X. Could be. This could be. I think that one might be it. Let's go back to it. B, the rate of reaction is 0. No. Just because the rate of reaction is plateauing does not mean it's reaching 0. It just means it doesn't increase anymore. So that's not right. C, the substrate has an inhibitory effect. No, that's definitely not right. The products have an inhibitory effect. No, nothing to do with that. So it is A. So to explain that, it's limited by enzyme concentration. Up here, remember, at this point, you know, if we've got sort of substrate molecules, let's draw them as little circles floating about like this, lots of substrate concentration, but there aren't that many enzymes. Let's draw them as kind of these little things here with their active sites. There aren't enough enzymes with active sites to kind of be breaking down all that substrate. So if you added more enzyme, you'd have more active sites available and the rate would increase. So under the conditions of more enzyme, the rate of reaction would continue to go up higher with more enzyme added. So it's definitely A. Question 5. Enzymes differ from inorganic catalysts in that they are highly specific. Which property of an enzyme is responsible for this specificity? Well, probably straight away you think, right, it's going to be the 3D shape. So this is a bit of a language question. Which one of these things means the 3D shape? And it's not to do the fact that they're insoluble. In fact, that's not even true. Enzymes are soluble proteins. They're globular proteins. I think all of them I can think of are globular proteins. B, the high molecular mass of enzymes. Well, that doesn't really, no, that has nothing to do with the specificity. This is the one. Surface configuration. What a horrible way to describe the shape of an enzyme, OCR board. Yeah, that is, the surface configuration just means shape. The shape of the surface. C. 6. What is the approximate temperature coefficient of an enzyme control reaction? This is always 2. It's worth saying that is below the temperature optimum. The Q10 coefficient is around 2. Above it, it's not going to be 2. 7. Which of the enzymes below catalyzes the following reaction? Sorry, that should be C, not 2. That shouldn't it? Okay, so we just need to know what type of reaction this is. Well, triglyceride being broken down into its constituent parts, glycerol and 3 fatty acids, that's a hydrolysis reaction. So therefore, it is going to be a hydrolase enzyme that does the job. Okay, here, 8. Got a little bit of text to read here. Got to fully understand it before we try and answer it. So, succinic acid dehydrogenase is the enzyme which catalyzes the oxidation of succinic acid during respiration. If malonic acid, this is the key part actually, if malonic acid is added to the system, the rate of reaction is reduced. Okay, well that bit there tells me that it's an inhibitor, okay, because it reduces the rate of reaction. But what type of inhibitor is it? An increase in the substrate concentration, succinic acid, increases the rate of reaction again. Okay, so immediately I know that it is a non-competitive inhibitor. So which one fits with that? Which one is a non-competitive? So I'll write that down just because that's my working at the moment. Okay, does it decrease the pH of the system? Well, I mean, it could do. I mean, maybe it does, to be honest, because it's an acid. However, this thing about adding the substrate and the rate increasing again, that wouldn't make any sense. So A is kind of a bit of a red herring, so I'll cross that out. B, it forms a permanent attachment to the active site of the enzyme. That is very unlikely. That is not the case, because that would be a non-competitive inhibitor. That would be something else. Okay, so that doesn't fit with the data. C, it has a similar molecular, there's that word again, molecular configuration. It has a similar molecular configuration to that of succinic acid. Yes, that's the one, because what it means is that the non-competitive inhibitor can bind into the active site temporarily, just like succinic acid can, and they compete for the active site. So they're similar in shape. So the answer for that one is therefore C. Coenzyme, no, that's not right. That's a red herring. Okay, 9. Here's a diagram. 3 curves. What are the 3 curves? One of them is with an uninhibited enzyme, one's with a non-competitive, and one's with a competitive. So you can either go through this and kind of work it out, or you can just label it here and then do that way. So this is going to be uninhibited, uninhib, okay, the full highest rate of reaction. This one is a competitive, because remember, with a competitive, we can get that rate going back up to the maximum if we add more and more substrate, and this is with a non-competitive. I'll just write non-comp. Okay, so which one is that? So P is the uninhibited, so that's those two, cross those ones out. And then the the competitive is Q, so competitive is Q, so it's this one. So therefore it's A. Okay, weird blank page there. Okay, 10. Which of the following is an extracellular enzyme? Well, how are you supposed to know? You're not supposed to know every single enzyme that there is, but you are supposed to know a few, and you will hopefully have heard of these. So catalase, that's H2O, takes H2O2 hydrogen peroxide and turns it into H2O plus oxygen. It's not balanced, you have to double this. But that is occurring in all, pretty much, living cells. So that's intracellular. DNA polymerase, well, what does that do? It copies DNA. Where do we find DNA? Inside cells, in the nucleus, in fact. So that is definitely an intracellular enzyme. Keratin, that's not an enzyme, that's a protein. Remember, enzymes are proteins, but they're proteins that do some catalysis. This is the protein in sort of hair and fingernails, in fact, as well. It's a structural protein, not an enzyme. Trypsin, now that you should be aware, hopefully you know this one, that is a digestive enzyme. So you should know that name. it's a protease, okay? It works in the small intestine. So I wasn't quite sure. Pepsin is the protease that works in the stomach, has a optimum pH of around 2. Trypsin works later in the digestive system in the small intestine / duodenum, and it has a higher pH optimum, I think around 7.5-ish. So that's D. Okay. If other conditions were to remain constant, which one of the following changes would explain a reduced rate of activity in an enzyme-controlled reaction? Okay, let's go through these one at a time. Increase in the concentration of end product, maybe. Okay, that could be a sort of end product inhibition. Increase in substrate concentration, no, that would speed up the rate of reaction. It wouldn't slow it down. Increase in enzyme concentration, no, that would speed up the rate of reaction. It wouldn't slow it down. Increase in temperature towards the optimum, no, that would still speed up the rate of reaction because it would be sort of going up the curve towards the optimum here, okay, being going up here. So the only one that it could be, if we rule the other ones out, is A. So yeah, might be. So could be with end product inhibition. Okay, so question 12. Which one of the following conditions is least likely to denature an enzyme? So can these things denature enzymes? Yes or no. High temperature? Yeah, absolutely, that can denature enzymes. Extreme pH? Yeah, that can denature enzymes. Maybe these two now you're less familiar with. C versus D. Which one is the correct answer? Well, both of these can reduce the rate of an enzyme, but they do so in different ways. So a low temperature does not denature an enzyme, but it just means that there's less kinetic energy, so particles are moving less, which means the substrate molecules are less likely to collide and enter the active site, collide with the enzyme and enter the active site, forming the enzyme substrate complex. But it doesn't denature the protein, it doesn't unravel it. Whereas C can denature an enzyme. So I found a paper, a bit of research here to show you that it can. If you wanted to go deeper into that, I'll put a screenshot in. But basically, the heavy metal ions can interfere with some of the ionic bonds or the disulfide bridges inside a protein structure, and they can cause it to misfold. And I think that is actually how nanoparticles of silver have an antibacterial action, just in case you're wondering. Okay, moving on. All right, moving on to 13. Which type of enzyme catalyzes the conversion of a dipeptide into 2 separate amino acids? Okay, this one's an easy one. This one is a hydrolase because it's a hydrolysis type reaction. Remember, condensation joining, hydrolysis is splitting. 14. The action of an enzyme depends on the specific structure of its active site. Which of these bonds are not important in maintaining the shape of the active site? So really any bond in the structure of a protein can be one that's involved in kind of stabilizing the whole structure and therefore controls to some extent the shape of the active site. So are these bonds in proteins? This is a bond in a protein, yeah, ionic bonds. This is a bond in a protein. This is a bond in a protein. This is not. So this is a bond in what type of biological molecule, can you remember? It is nucleic acids, DNA or RNA, so the answer is D. 15. The diagram below shows some reactions in a kind of metabolic pathway. So we can see an amino acid is converted from 1 to 2 to 3 by enzymes V and X. When an excess of amino acid 3 is added into the bacterium, the rate of the reaction is reduced. What is this an example of? So 3 probably comes over here and inhibits V. So this is end product inhibition. 16. Okay, a sample of starch suspension was tested with iodine and the result was a blue-black colour. A second sample of the starch suspension was tested with Benedict's reagent and the result was a blue colour. Well, that's because Benedict's reagent is blue. So Benedict's reagent is blue and it just basically stayed blue. The third sample of starch suspension was mixed with amylase, okay, and incubated at 30 degrees C for 10 minutes and then tested with burette reagent. Okay, this is a trick question here. So this is still going to be a blue, a blue colour. So it's still going to be blue because amylase tests for, burette tests for Oh, hang on a minute. No. Wait a sec. Okay, almost got me there. So burette reagent is a test for proteins. So it's going to go purple if proteins are present. And in this one, it's tricky because it's not thinking about the starch. It's thinking about the actual amylase itself. Amylase is an enzyme. It's a protein. So therefore, it will, with the burette reagent, make it go purple. Nice question there. Almost got me. It's D. Okay, so 2 questions about this graph. So, oh my gosh, we got a lot of different lines here. Initial reaction rate, substrate concentration. Okay, which one of the curves, A, B, C, or D, represents the results when the same experiment is conducted in the presence of a fixed low concentration of a reversible competitive inhibitor? Okay, so X is normal. Reversible competitive inhibitor is going to be A. Okay, competitive inhibition, remember it can reach that top line. And it kind of follows a similar curve just underneath the first one. Which one of the curves represents when the same experiment is conducted in the presence of a fixed low concentration of an irreversible non-competitive inhibitor? And this would be B. When an enzyme is subjected to temperatures above the optimum, it denatures. Which of the following bonds are the first to be disrupted by high temperatures? So the first ones are the weakest bonds, okay? So let's rank them. Let's go from strongest to weakest, okay? The strongest bond probably is the peptide bond because it's a covalent bond, and so is the disulfide. So those are both very strong bonds. Ionic bonds are also very strong, and hydrogen bonds are the weakest, okay? So it's B. Final question, on to 20. Which one of the following graphs shows the effect of pH on the reaction velocity of a typical enzyme? Note in each graph the scale of pH is from 2 to 13. So typical enzyme, that's an easy one. It's this one right here, okay? B. Brilliant. Okay, so that's the end of this video. Looking at those enzymes questions. Do make sure you try and learn from any mistakes that you've made because these questions do get repeated quite often in exams. Thanks very much, and I'll see you next time.